everyone, I'd like to welcome Dr. Steele. He's coming to us today from the Yale University School of Medicine, where he's an assistant professor, and he's well known, I think, to a lot of us from his work that he's done with TMS at Root Center. He earned his bachelor's degree and Ph.D. from the University of Minnesota, and his Ph.D. is in cognitive and biologic psychology. So today he's going to be talking about transcranial magnetic stimulation and substance use disorders, and we're super thrilled to have you. So I'll hand it over to you, Dr. Steele.
Well, thank you very much for that introduction and the invitation. I really am looking forward to being here today to share some of my recent stuff. Let me see if I can get the screen share to work. All right, unless I hear otherwise, I'm going to assume that worked.
You can see it.
Okay, great. Yes, so I'm here to tell you something about what TMS is, transcranial magnetic stimulation, and then where a little bit of a background, how it might be applied for substance use disorder, and then where I think we should go, and where my lab is going, and how we interact with you all at the Root Center. Let's see, click the right thing. There we go. I start with where my funding comes from. I'm in Yale Psychiatry, and my lab actually is in Hartford Hospital, so I have a Hartford lab. I've gotten funding from NIH, from National Institute of Drug Abuse, and then the Center of Compulsive Behaviors. And then also most recently started this week, I just consented my first person from HJ yesterday for a new grant that came online, a NARSAT grant. I also like to get this out of the way because I don't want to miss anybody. There are lots of folks that helped me get to where I am today. And so this is just a short list. And then some of you already know my lab members, Sebo and Handshake out. They're instrumental to the work that I'm doing and as we move forward. Also, I'm not making any money anywhere else, so the declarations are that. I should also like to start with, for this group, I'm a scientist, academic researcher, so I'm a big nerd. I play with really big, expensive, sort of complicated toys. I want to tell you something about each of those today. I sort of get in the weeds sometimes. Don't come with me. Just think about the top level main points that I'm trying to make. And then if you have questions about what the weeds are like, ask them, jump in, or let's find another time. Email me. I'm happy to answer questions, talk about TMS research, how it applies to what you all are doing at Root Center. And then I'm also open to giving lab tours. So if somebody wants to come and see the fancy toys, come on by, let me know. All right, well, so with that, let me tell you about what TMS is. So this is just a depiction of TMS coil. It's a figure-of-8 coil. As you can see, it has a figure of eight there. There are just copper wires in there that we pass electricity through quickly, and turning it on quickly on and off generates through electromagnetism, magnetic pulse underneath, and then an electric current underneath that. But then, hopefully, when you put it near the brain, it can fire the neurons or the cells in the brain that are related to function in the brain. And then hopefully then also, you could use a small spot in the brain as a window into areas that are beyond that small spot. So we get this small focal stimulation of neurons that are connected to other parts of the brain and hopefully change behavior and hopefully rewire the brain. How it might work in substance use disorder is, again, we would apply this in the frontal part of the brain. This DLPFC is just frontal part. Think of it as like executive control, top-down control, how we control our behavior. That is connected then to downstream regions that are related to reward and then dopamine release. So there's a potential by stimulating in the cortex and the surface of the brain that we get downstream projections and maybe change reward processes, change dopamine processes. All of these are related and implicated in substance use disorder. I use a figure-of-8 coil. There are different styles of coils. So they started with the circular coil and then the figure-of-8 coil. And we also have this thing called an H coil or helmet coil that can get deeper in the brain. Sort of the distribution of the magnetic field for a circular coil is sort of diffuse and superficial, so it doesn't go very deep. It's another one that stays right on the cortex. See, the figure-of-8 can get quite focal. So that's why I'm saying we get a window of stimulation in a small part and hopefully just modulate one thing and not several things. The deep helmet coil is very diffuse. It does get deeper in the brain, but it stimulates all the neurons on the way down. Pros and cons to all of these. I like this one for research because I'm interested in what the brain does, and I try to have a focal stimulation point such that I know if I change something, I know where I was and what I changed, and then I can measure it. This is an example of a TMS setup. I own this bag vector one. This is the one you guys have in Middletown. You have this chair. There's a couple coils, stimulator at the top. And then this is a cooling system to make sure that the system doesn't overheat. There are a couple Turner knobs on here. And then the big question is how do we know how high to turn up TMS when we apply it for intervention? Well, we need to figure out the dosing. So this is just another depiction of a TMS coil over somebody's head. We start by stimulating the motor cortex that hopefully moves this finger, right? Turn the coil up and down. A depiction of this, then again, the electric field fires neurons in the brain that are connected to muscles in your hand. That then generate electric current that we measure in that muscle. So each line is where I've given a single pulse that I measured here of activation in the muscle to move the finger, right? And so as you can see, as I turn it up and down, the peak of this goes up and down. So based on that, I can identify what somebody's dose is, how much energy do I need to put in for my TMS system to stimulate neurons in the brain, in their brain on that day at that moment. Everybody's dose changes over time, and everybody's dose is slightly different over time. So it's important to measure that. Once you have dose, we can move off the motor cortex and move to the front part of the brain, where I think is a good intervention point. And then we would call a treatment. So repetitive transcranial magnetic stimulation, or RTMS. There are different sequences. So instead of just giving a single pulse over the motor cortex, we would give repeated stimulation. So here is a repetitive 1 pulse per second. So it's a low frequency stimulation. We call that 1 hertz stimulation. And then if we did 10 pulses per second, so it's much faster, then it would be 10 hertz stimulation. They're both types of RTMS. One is thought to be inhibitory in that it sort of turns down the ability of those neurons or those cells to fire. It makes it harder for them to fire. Or you can turn them up, so it's easier to fire. So you can maybe increase or decrease the connectivity and the working of that circuit you're targeting based on if you do different sequences of RTMS. Quick sidebar on nomenclature. TMS is sort of this umbrella term. All TMS, RTMS, TMS, all pulses are TMS. Largely people use TMS to describe just single pulses when you're finding the dose. But technically RTMS is a type of TMS. RTMS describes all sort of repetitive sequences. I just talked to you about 1 hertz and 10 hertz. We'll talk in a minute about ITBS and CTBS. Anytime you get into a pattern stimulation, then it's an RTMS type of intervention. And those are the ones we use to intervene for clinical population. Okay? All right, so back to how do we know that they're excitatory or inhibitory? Back to this motor evoked potential. This is the motor of moving this finger. We apply TMS. We measure that before TMS. We give either a sham TMS, a 1 hertz or a 10 hertz TMS, and then we measure motor evoked potential again. So sham, it didn't change. 1 hertz, it reduced, and 10 hertz, it went up. So inhibitory, excitatory. So what's happening in the brain other places besides the motor cortex? Because we'd likely move out of the motor cortex for intervention. So we move forward in the brain again. Here, same thing. Before measures, there's just a brain activation map. We do sham, 10 hertz, 1 hertz. Sham didn't change. 10 hertz actually increased activation in that part of the brain where they're stimulating. And then 1 hertz decreased stimulation or decreased activation in the brain after stimulation. Okay, great. We have some idea of how it might work. If we apply it to a clinical population, what might happen? About the same time we were identifying that it's excitatory and inhibitory, Mark George and his group was applying this as a treatment for major depressive disorder. So here they have folks in treatment for MDD. At baseline, their depression scores are standardized. And some of them are randomly assigned to get sham TMS first for 2 weeks, and some are get active TMS for 2 weeks. The active group, their depression scores went down. After a slight dip, this is probably just a placebo effect in the sham, their actually depression scores went up after 2 weeks of not getting any kind of active TMS. And then they did this crossover design, which is actually quite powerful to show that it's not a placebo, it's not a treatment intervention effect, it's actually TMS that's changing things. The folks who were in sham first, but then active, their depression scores decreased once they got onto the active TMS. Folks who were on active and switched to sham, their depression scores went back up, suggesting that the TMS intervention was actually efficacious in treating depression in this sample. Since this time, this 10 Hertz stimulation, that's what they were using, has been FDA approved to treat depression. Again, they applied over left DLPFC in the front part of the brain. They were using a 30-minute sequence. There's now a 3-minute sequence that I'll tell you about in a minute. That's also been FDA approved. It takes 4 to 6 weeks to get the number of treatments in, and roughly 25 to 30 sessions before you see clinical efficacy. RTMS is FDA approved to treat MDD, like I was describing, but also obsessive compulsive disorder, and then this TMS is applied to treat migraines. Recently, these are all figure-of-8 coil stimulations or indications. The head H coil, deep coil has been recently FDA approved to be an adjuvant to other smoking cessation interventions. So TMS seems to be effective for clinical populations. What if you don't want to do 30 minutes? There are other sequences. There's this CTBS, it's continuous theta burst stimulation, or ITBS, which is intermittent theta burst stimulation. And now we've got continuous is 40 seconds of that. ITBS is 2 seconds on, 8 seconds off for stimulation, is after 3 minutes. And we've identified that CTBS, like 1 Hertz, is inhibitory. ITBS, like 10 Hertz, is excitatory. The big change is that here, each spike is a single stimulation. Here, each spike is a triplet of stimulation. So if we blow it up, you see each of these spikes is actually 3 stimulations, repeated very quickly. So you get a lot of pulses in a very short period of time to get inhibitory stimulation or excitatory stimulation. Okay, great. Shorter may not be better clinically. Could this be the same as an ITBS intervention? The folks in Toronto do a lot of this in clinical populations. So they compared a non-inferiority study at 10 Hertz RTMS versus ITBS in treating depression. Depression scores at baseline over the treatment, 5 weeks of treatment, and the treatment and the 1 week follow-up. They were the same. ITBS, 3 minutes, 30 minutes of 10 Hertz RTMS proved effective in treating this clinical population. So that's sort of the background of what TMS is, maybe how it's used clinically. It's FDA approved for depression, so there's a lot of work out there highlighting its efficacy there. But we're all here treating depression, or not depression, but substance use disorder. So what does neurologization or RTMS do in substance use disorder? So,
Yes.
okay, so once again, we're hitting the front part of the brain, this executive control process, and then this sort of reward process downstream. The idea here is that there's accelerator and a brake process that is dysregulated in users. And these two processes are related to top-down control with the brake, and then the reward processing is the accelerator. Which one is dysregulated? Is it a combination of one or both? Is the accelerator stuck, or is the brake can't control the accelerator? Or is it a combination of the two? With TMS, we could be able to hit both, one or the other, and turn one up, turn one down. Okay. Some ways to do this in animal models, they have a similar effects. This is a mouse model. They have a part of the brain that says analog to the same frontal part of the brain in humans. They have the same, very similar reward circuitry, dopamine release circuitry that is targetable with TMS or neuromodulation in this case. So what we do here, in the mouse models, they do brain surgery and they essentially put a laser in the brain of the mouse, pointed at a specific part of the brain, and they turn it on and off, and they put different wavelengths of color and light in there. And that can either be excitatory or inhibitory. So you can see how we can't do this in humans, but it's also a really good model to be able to cycle through a bunch of different types of interventions to say what sequence, excitatory or inhibitory, what part of the brain can turn on and off the accelerator and the brake. So an amazing mouse model that was published about 10 years ago did this. Now, not all preclinical models or animal models are created equal. Not all are really, really good models of human addiction. This one I'm going to step through a little bit to highlight how it is a better model of addiction. So what they've done is they've given mice access to look for cocaine in their cage. They get a lever, they press a lever, they get an infusion of cocaine. They get 30 infusions a day for the first few weeks. Then they get up to 80 infusions a day for a couple of weeks. And then they go back to how much are you going to take once you've been exposed for a couple months of cocaine. Now, this is where most models end. If an animal, human included, is put into a box and there's one lever, and that lever gives you cocaine, you're going to press it. You're going to use cocaine. Just because you do that doesn't mean you have an addiction. Doesn't mean you have substance use disorder. So that's not a great model. Humans do this too. I would do this, you would do this, because there's nothing else to do. It's just what you would do, right? Now, some humans, the folks that you guys are treating, and that I want to see and try to develop a treatment for, go through adverse events and continue to use. So the adverse event that they're using here is they're electrifying the floor. In order to get over to the lever, the animal has to get a foot shock, and then they get a lever and they get a cocaine infusion. So in known negative consequences, in the face of known negative consequences, these animals have to go through that to get cocaine. As you might expect, there are many rats, or mice in this case, that use cocaine. They all use cocaine in this open seeking behavior. As soon as they start getting shocks, a large number of them stop using cocaine and say, you know what, not worth it. I'm not doing that. And a lot of us are like that in society too, right? There are some people and some mice who are resistant. They say, you know what, I like cocaine enough, or I have brain circuitry that drives me to use it. I'm still going to use. And you can see that the number of lever presses just stops for those who are sensitive, but retains for those who are resistant. So this is number of infusions, or number of lever presses, number of infusions tracks the same, and the amount actually they get, there's a reduction in use when there's a negative consequence related to using cocaine, but they're still using cocaine at a very high level. This group mimics human addiction. This group is the one we're interested in. When we stimulate that group, can we change their cocaine-seeking behavior in the part of the brain that's the accelerator connected, or the brake connected to the accelerator? And in fact, they show that they can reduce it. They don't eliminate it, but you can reduce cocaine-seeking behavior by stimulating the part of the brain. So that was a decade ago. Since then, what does this look like in humans? It sort of opened the floodgates. Like, we should be trying this in humans. It seems safe. There's some safety precautions we have to think about. But with a single session or acute application of RTMS, we can change pre-post craving, self-report craving in all drugs of abuse. Marijuana should be on here. That's also on there. Almost any drug of abuse, we can reduce it by hitting the same dorsal prefrontal cortex and brake connected to the accelerator. Chronic applications, this is just more than one RTMS session in a day or over several days, also reduces craving. This suggests that RTMS could be an intervention, a promising intervention for substance use disorder. I've been talking a lot about cocaine use disorder, primarily because there's really good preliminary data, a bunch of that preliminary data is in cocaine, but then also there's no FDA approved treatment for cocaine. So this is a big problem. We need to figure out how to have a good treatment for people who are suffering from cocaine use disorder. That is not to overlook the opioid overdose crisis that we're dealing with, layered on top of this pandemic. That just made it completely worse. We need to develop better treatments for folks suffering from opioid use disorder, cocaine use disorder, and the like.
She never
One of the struggles
digs in the trash, never has the trash and found an edible that I threw away.
I think Simone,
All the doctors, the edible was pretty, there was no chocolate in it or anything. So she threw up a little bit, said,
moving on, I guess. The rest of
Somebody
Oh,
mute
sorry about that. Yeah. Can we mute?
but
Simone is unmuted.
Thank you.
Yeah, thank you very much. Right, so I think we should use, well, we should think about how we're assessing substance use disorders, and this may be the problem, why we haven't been able to develop an effective treatment to help everybody. One is we use urine samples. They're easy to get, they're quantifiable, but you know what? It tracks the behavior of the disease and not the disease, right? You can go out and you, have a couple drinks of alcohol and come in and test positive, but that doesn't mean you have a disease, it just means you used, right? It's different. Similar with craving. Craving is easy, it's self-report, but it's subjective and it's actually not obligatory. You don't have to crave to relapse. So it may not track exactly what we want. You can get into a deep dive in clinical characterizations, which may or may not be helpful, but often they track behavior as well, not necessarily disease. The example I like to give here is that we're measuring symptoms of the disease and not the underlying disease. So when you go to the doctor and they give you antibiotics, they say, take the entire course of antibiotics. Why does the doc tell you that? Because she knows that whatever you're treating, the infection, the symptoms of that infection could disappear before the underlying disease disappears. So if you stop treating the disease after the symptoms go away, the underlying disease is still there. We need to attack the disease. So I think the addiction is a biological brain disease, chronic relapsing. We should be measuring things in the brain. I do that with electroencephalography, EEG, and MRI, magnetic resonance imaging. These two get at different but sort of comparative areas of the brain. EEG measures electrical activation in the brain. All the cells in your brain and your body really move on electricity, and we're just measuring those in the brain. In order to fire neurons in the brain and use and develop electrical impulses, you need oxygen, so then blood needs to go there, and MRI measures the blood. So these two are sort of measuring similar things, in that we're measuring when and where something is happening. So with those, I think we should be using brain-based substance use disorder interventions by measuring the disease where it lives in the brain. What does that mean exactly? Where in the brain is it? How do we identify where that is? This is just an example of areas in the brain that are part of the mesocortical limbic dopamine system. This system is related to reward dopamine processing. We know that it's dysregulated in users versus non-users. This is just showing that cocaine users versus healthy controls, these areas in the brain don't communicate as strongly or as well as healthy controls. Showing that these are regions that we know are dysregulated and implicated in cocaine use disorder. It maps on really well with the brain disease model that's put forward by NIDA and NIAAA, the heads of those two institutes, where it shows this cyclical model. Many different regions in the brain are related to it. But it's important to note that there are different stages of this disease. People, when they come in for intervention, may be in one of these disease states. They're acutely intoxicated, they're in withdrawal, or they're craving, something like that, one of those regions. Each of these parts of the cycle implicate different parts of the brain as to being important to processing things while they're in that state, right? That may show that certain parts of the brain are malleable, open for intervention, depending on what state somebody's in. Also, we know that this maps onto cognitive functions, executive control processes, cue reactivity processes, affective processes. Different parts of the brain are differentially affected depending on what state somebody's in and what area of the brain is being targeted with specific cognitive functions. Also, this at the bottom here talks about neuroadaptations. This is talking about neuroplasticity. The brain changes over time. This is how we develop memory. You learn new things by the brain changing. There's neuroplastic change. It's the same for exposure to drugs. The first time you're exposed to a drug, you're not addicted. You have to go through this cycle several times and your brain changes, becomes more and more dysregulated. I mean, some people may be more disposed to that, but they're not addicted until they start using, and they use multiple times. So the idea is that these dysregulated circuits have neuroplasticity that help them get in. Perhaps we could use neuroplasticity to help them get out, right? We could identify these or target these dysregulated circuits in a way that could benefit individuals in the long term. So then I guess the question is Are all these circuits, are all these areas worth targeting? Where do I place the coil? How do I do this, right? I started by trying to answer this question with outlining 6 known unknowns. I put out a review and commentary last year with the student in mind, talking about 6 areas that we need to target and better understand with TMS. We need to understand, where do we stimulate? What part of the brain, right? I talk about accelerator and brake. I think that's a good place to start, but why, right? We need to measure things downstream in making sure that we're engaging and modulating those targeted circuits, right? We don't really know if we should hit with excitatory or inhibitory. We don't know how many sessions we need to apply to have long-term change. I think TMS should be an adjuvant to other treatments. So like you guys use methadone and buprenorphine. I think that's a good place to start. I apply TMS on top of individuals who are already getting medication-assisted treatment. We need to worry about the brain state that somebody's in. Remember the cycles. Where are they in that cycle? We need to standardize that, because certain parts of the brain may be malleable depending on where they are in that cycle. And then I want to use primary outcomes. Diseases in the brain, we should be measuring the brain. And that's primarily where I'm going. We're still at, where do we start? What part of the brain, what cognitive functions, right? So I've done a lot of stuff with inhibitory control. I have this go, no go task. Very briefly, individuals are presented individually, just X's or K's on the screen. They need to make a response when they see the X. Don't make a response when you see the K. It's a go, no go. Make a response when you see it. Don't make a response. No go when you see the K. There are many more X's than K's, so it develops this prepotency to make a response. So individuals just get into this process. So it makes it harder to inhibit your response when you see the K. Also, it doesn't really, so that's response inhibition or inhibitory control. Everybody makes a mistake. Everyone presses a button when they see a K at some point in this task. What do they do with that information is very important. Very much different between users and non-users, this error processing. So what I did is I collected EEG during a go, no go. Don't worry too much about the specifics here, other than onset of the error is at 0. There's initial negativity. Is it processing of an error? This is Homer Simpson going, don't, I made an error, right? That's what that one is. The secondary process is, okay, I made an error. What do I do with this information? Next time I see a K, don't press the button. There you go. That's what that one is. So there's sequential processes that are differentiable with ERPs. Now, I measured this with individuals at the baseline before they initiated drug treatment program. Some of them subsequently completed in the red line. Some discontinued prematurely. Just visually, you can see they process errors differently. They have differential processes on this initial. They have differential processes in the secondary. With machine learning models, just some fancy math, I can look at people who ultimately completed drug treatment versus those that discontinued drug treatment. Can I actually predict it based on their brain measures? And in fact, my prediction models, I predicted those who completed at almost 80% rate. And I also predicted those who discontinued prematurely at 75% rate. These are really high numbers for these types of models. It's important that both boxes are red, and then the gray boxes are the error. So the ones I missed in prediction. Also with this method, I could identify it's a secondary process, this top-down control. What do I do with this information process? That if I enhance that, I could actually increase maybe how long somebody stays in treatment, right? I have a cognitive process I can target. What part of the brain is implicated in this? Here I've got the same task in the MRI scanner, and I've got medial frontal areas down to the mesocortical and the dopamine system areas that we know are dysregulated. And their communication between those are stronger in those people who discontinue or complete relative to discontinue. So if I can strengthen this in individuals, they may stay in treatment longer. They may have longer, better long-term outcomes. Again, I can do a really good job of predicting outcomes here. You see the red boxes are also very high. So this gives me a target, both cognitive function, the area that I want to modulate with TMS. And so this is what I did when I was in Baltimore. I applied TMS to this part of the brain to try to increase somebody's ability to either stay in treatment or actually, in this case, reduce cocaine use. So what I did is I developed a proof of concept study where I recruited 10 cocaine-dependent individuals. I did open label, so everybody got active TMS. I applied ITBS, this excitatory TMS, to the left side of the brain, left part of the brain. I did it 3 times a day for 2 weeks, so they got 30 sessions. While they're doing TMS, I showed them cocaine cues and try to engage the circuit, try to get them into a state of craving, and then I want them to downregulate their craving. With the idea that with excitatory TMS, I can increase their ability to downregulate their drive to use cocaine. And then I had them come in 1 week and 4 weeks later, see how they're doing, what their drug use behavior has changed. The main questions were chronic RTMS, can I decrease cocaine use? And then this is very similar spot that they use to treat depression. So do other things change as well, like mood, other drugs of abuse, things like that. Long story short, both amount and frequency used. I can show you that at baseline, the bars are the averages across people, and each dot is an individual. So amount used and frequency used at baseline for the 1 week and the 4 week follow-up, both of them decreased across people. Everybody reported a decrease in use. Nobody stopped using. It was good harm reduction. But again, I don't know how many sessions I need. And I don't know if I tuned it perfectly for each individual. Obviously I didn't because nobody stopped. But everybody reported a different interaction with cocaine. They couldn't get as high when they did use again. They didn't have the same drive to use as they did before. They could start and stop. So their compulsive use actually reduced, if not stopped in some people, right? So that's all great news. The other side effects or off-target effects is if they use other drugs of abuse, like marijuana, alcohol, and nicotine, those also tended to reduce. It's a small number, so it wasn't significant, but it seems it's the same circuit. So it's quite possible that I'm moving everything all at once, even though I'm talking to them only about cocaine. Everybody's mood picked up. Nobody was depressed, but they all got a little more pep in their step day 3, day 4, which is pretty impressive. So there are these off-target effects that also happen, seem to be quite positive, right? So long story short, the reason I did this first study was FDA and my IRB in Baltimore was very concerned about safety. Now, a quick sidebar on safety. TMS, if applied incorrectly, outside the safety parameters in people who are high risk, can induce a seizure. The most significant negative side effect is a seizure. Now, that is exceedingly rare in people who are properly screened and not high risk. Almost all of the TMS-induced seizures in the last decade or so have been in people in high risk or beyond applied TMS in beyond safety range. Cocaine is one of those things that can increase safety risk. I designed this study for somebody who came into the office and said, you know what, doc, I need something today. Instead of the doc saying, you know, I can get you a bed in 2 weeks if you can stop using. I want to say, well, you know what, I have a TMS in the back room. We could do some sessions today to get you on the right track. So they would test positive. Metabolism, cocaine is quite quick. So as long as they're not acutely intoxicated, then it should be fine. But the FDA was very concerned that maybe I was going to induce a bunch of seizures. Suffice to say, I didn't. Safety is very important. The most common side effect is you feel pain underneath the coil, and I could induce a headache that usually goes away on its own. So really, TMS in the right hands with the correct safety screener is quite safe, as long as we worry about people who are at high risk. So with that sidebar, did I decrease cocaine use? Yes, and there are some side effects, off-target effects as well. But everybody knew they were getting active TMS, so there's high potential for placebo effects here, right? It's a small n, so it's hard to make really firm conclusions about this. And then I didn't get any brain measures, so I don't know what I did in the brain to change it, right? So what we need to do is what I did next is I developed a clinical trial that I set up and started running before I left NIDA in Baltimore, what we call an expanded feasibility study. That's the FDA wanted that language. It's a double-blind, placebo-controlled RTMS treatment to reduce cocaine use. And I want to assess neuroplastic change. So it is the same 2 weeks of treatment. I recruited controls as well to better understand the neuroplastic change in just acute stimulation for healthy controls versus users. I added MRI scans at baseline to contrast users and non-users and get acute differences of change in the first day of MRI scans, and then longitudinal MRI scans to better assess how long things last in the active TMS. Contingency management is just a monetary reward for continuing to provide negative urines. Everybody got that. Some people got active TMS, some got sham TMS. Now, I set this up before I left, and then COVID hit, and then the throughput of NIDA is really quite slow. So this is going to be a long-term coming. COVID has really slowed it down. But this is designed as sort of a definitive trial for cocaine use disorder and understanding the neurocircuitry that changes with TMS, with my best knowledge from about 4 or 5 years ago. Now, where can we go now in my life? I'm interested in understanding the neurocircuitry. What's malleable? How do I measure that? How do I change it? How do I sort of titrate this to tune it so that it's most effective for most people? So what I'm doing is I'm recruiting healthy volunteers from the community and then cocaine or opiate users from treatment centers in the area. While treatment seeking, I want to do this as an adjuvant because they're currently in some sort of group or they're on MAT. I'll measure EEG or MRI when I get brain signals to get state trait characterization so I can compare users and non-users, just at baseline. I apply then an acute session of RTMS, both active and sham usually. EEG has an ability to get simultaneous EEG and TMS. I have those capabilities in my lab. And then measure this again, the same sort of cognitive functions. So I'll focus on executive control, acute reactivity, before and after an acute session of RTMS. I can get a pre-post, a state trait, get an active versus sham TMS manipulation to understand what's changing where, how do I place the coil, which circuits are malleable, how do I target it most effectively to induce change. So I'm interested in which of these dysregulated circuits in addiction are malleable that I could scale up to a treatment. I think with an acute session, I'll be able to identify those so that I can scale it up and be most effective in long term. Machine learning is just how I can predict future outcomes, who will find this effective, who will develop positive outcomes for treatment. That's all with that. So I'm going to go back to these 6 areas of known unknowns. How am I addressing those? I talked to you about a lot of these in the last 40 minutes already. How do I identify where to target, where to stimulate my TMS? Well, I use previous data that I have to identify targets with cognitive function and the neurocircuitry that's downstream that I could target with an intervention. How am I measuring circuit engagement? I use EEG and MRI measures that I talked to you about. So I use executive control tasks and cue reactivity. How do we optimize our TMS sequences? That's actually what this study is that I just started this week. I'm comparing excitatory and inhibitory stimulation at the prefrontal cortex to assess if I'm moving the accelerator or the brake in opiate users who are on methadone. Is one more effective than the other? Modulating the circuit. Is it an effective way to intervene with individuals? Right. Again, I don't think RTMS is a standalone silver bullet, so I recruit folks who are in treatment already. I think that's going to be the most effective initial application of RTMS. I try to engage the brain state, in this case by having them look at cues and downregulate their craving for those cues to try to increase their ability to decrease their craving in the future. I'm interested in primary outcomes. So again, I look in the brain. I can use behavioral outcomes that are related to brain-based measures as well. That's a general outline of how I sort of tackle TMS studies as an intervention for substance use disorder. I want to highlight at the end here just two of these areas that overlap with exactly what you all are doing as well. So one is number 2 and one is number 4. So subcortical circuit engagement. I do pre-post MRI with an acute TMS in the middle. I have a small pilot grant from Harvard Hospital where I'm doing this I recruit opiate-dependent individuals that are treatment seeking, primarily from Henderson Johnson, Kim's Health, HJ, right? They get active sham ITS. I'm interested in this executive control process, the cue reactivity, and this pre-post measurement, right? With the big question of which of these circuits are malleable. With 10 people, I should be able to get an idea of which circuits are malleable in imaging, and then sort of move from that and plan accordingly. Either I scale up to a larger study or I do a different location because I've identified a different place that would target the circuit a little bit better, things like that. I do this in my lab in Hartford Hospital, the Oral Neuropsychiatry Research Center, in collaboration with you all at the Root Center. Roots are not advanced. There we go. So I had folks come, and this is part of a lab tour. So you can see Kim and Ray and Eliner came, and I showed them all my fancy tools. They're also helping me recruit. So I very much appreciate it. But without their help, this project wouldn't be going as well as it is, and my new study wouldn't be either. A study that I haven't started yet, that I want to do, this is in the future of my lab, that I'm working on, is a chronic application of RTMS. Once I have some idea, I already have some ideas of where to place the coil, obviously. Once we get some ideas better, potentially optimizing where it goes, we could try this as a behavioral intervention, where I apply this in a larger group of individuals, opiate-dependent, treatment-seeking. Again, RTMS is an adjuvant. We could just do open label, everybody gets active. Maybe we add sham in there too, but they get chronic excitatory TMS. Here, with this long-term longitudinal measure, I'd be interested in actually changing behavior. I think that acute measures change something very quickly, not very long, maybe an hour. And then the chronic applications could have longer lasting. Like my cocaine stuff in Baltimore had at least 1 month of change, maybe longer. We didn't measure it any longer. But the clinical trial that I set up had a 6-month follow-up, so we would have some idea there. So here we'd like to know how long things change, what other off-target effects, other drugs of abuse, other mood disorders as well. And so is behavior malleable with a chronic intervention? I think it is. It's just a matter of doing the work to identify how best to do it, right? We could also add imaging there and better understand what's happening in the brain, neurocircuitry, right? We get pre-post measures on the long-term application, and then we get at neural changes that are related to executive control, cue reactivity, or relate that to behavior. Which of these neural changes are related to behavioral changes? And so these are two sort of small pilot-ish studies that would take a lot of effort to complete, and would really give us good insights as to how RTMS could be tuned in perhaps an effective treatment for an intervention. They're both scalable for FDA indication. So we could work towards developing RTMS as an FDA-approved treatment for substance use disorder. So with that, I just want to say thanks to everybody. Nobody jumped in with questions, so hopefully you saved them all till the end. Again, send me an email. I'm happy to answer questions beyond this time. Or if you're interested, let's find a time I can give lab tours and come see the fancy tools we can do TMS together. With that, well, thank you for your time.
So Dr. Steele, I have a question.
Yeah.
Is there an unintended consequence of using TMS where people might become less depressed? And if so, is there any implication with the results of your study with the decreasing cocaine use? Or is there any factor that might be
it's difficult to know, right? So I did a lot of talking, and a lot of the field is talking like this is a cocaine user, but people are super complex, right? You can't just change one thing in isolation. You're going to change the person at some level. So it is important to understand the person as a whole and how their mental health is related to their substance use disorder. And that's why I advocate measuring all those things. A lot of folks don't measure everything because we don't know if it's in depression. Most people who are depressed also smoke cigarettes, right? Or nicotine in general. But most clinicians who are treating depression don't measure that. And so it probably all comes together. But we don't know because we don't ask those questions. It's an interesting question about as complex humans change their behavior and we have an intervention, what else changes and what does the interaction do? It's important and yet unknown.
Thank you. So any other questions? We kind of open it up to everybody here.
Oh, hi, Dr. Podolski, Dr. Steele.
How you doing?
I did have a question, Dr. Steele, about the study you did with the cocaine users.
Yep. Did we lose Gabby?
Sorry. For the cocaine users, you said they didn't completely stop using, but were they also in another like treatment program for cocaine use, or no?
No, none of my folks in Baltimore were what you would call officially treatment seeking, and they weren't in treatment in general, right? I think it's a long shot to say that any single treatment is going to fix any single person. And so I think it's easier and more reliable to recruit folks who are already in the mindset to reduce their use, already on the regimen, and then we just apply something on top of it to help some people get over the hump. In this case, we were testing an experimental treatment, and everybody was interested in maybe cutting down if it worked, but it wasn't their main focus of coming. And they weren't
Right.
Yeah.
Oh, well, I was just suggesting that maybe if they were in an additional like treatment program for their use, maybe it might have ceased altogether. Like
Yep.
the idea you had with your RTMS, I think that would be great, like in combination with the treatment they're already receiving at Root Center.
Yeah, so we in Baltimore, they reached out. I tried to get into treatment centers there too. They're not as friendly. And we were going to partner with them with a HIPAA release form so that we could talk to their clinicians to talk about how to titrate. We were focusing on buprenorphine because it's a little bit more stable than methadone, less liability for ODs and diversions and such. Right. So in chronic applications, I think you would have to make an effort to assess necessity of changing dosing. Because if you're changing mechanisms in the brain, that may change the necessity of specific pharmacological intervention too. And it's an unknown question. In the acute TMS that I'm doing now, I don't think I need to worry about it because it should last about an hour and that's it. I don't expect any kind of long-term change. I see Dr. N asked something very similar about methadone dose changes after TMS would indicate positive benefits, which I would agree. So I think long-term change, long-term chronic applications of RTMS in relation to understanding what their physician suggests for methadone and buprenorphine levels could be paired together as a long-term treatment. Yep.
Yeah, I think that would be interesting to kind of pair that with your research and see how those levels maybe taper off over time.
I think it would be essential if you're going to do a chronic application of RTMS in a treatment group that's already on a pharmacological intervention. You have to keep track of what their dosing is to make sure they're on enough, but not too much, right? Trying to look at the Dr. N asked extinction times for these positive effects. So the question, as I interpret it, you could put it back in the chat if I don't get it right, is how long do effects last? It's a good question. Unsatisfying answer is we don't know. We think that a single session of RTMS changes the brain for about 1 hour, which is why I try to put somebody in a scanner right away after I do TMS, measure the brain, and then they go home and they're unchanged. We don't know how long the intervention lasts, depending on how many sessions they have. I can say one of my people that was in the 2 weeks of intervention is actually a real success story. She's one of the ones that highlights that she could decide not to use cocaine, but instead you spend time with her granddaughter. She'd gotten a job while she was in treatment. We started with inpatient, so she got to a negative urine. She could go get the drug test, get the job, got the job a week after she got out of our inpatient. I saw her a year later. She'd been able to retain that job. She left with reducing her use quite a bit after the 1 month follow-up. and she'd retain that use level. Same interaction with cocaine. Now it's all self-report, so it is what it is, and it's a single person, so that is what it is. But it seems quite promising. So my job is to try to figure out what it is, how to tune it, how to target it a little better, how long does it last, how do we make an effective treatment, scale out to as many people as possible. So hopefully that answers your question. I think the other ones in the chat are just comments. Anybody else have questions?
I just had a comment about the success story you were just telling. I think it's interesting how she had the negative urine test and then got a job. So that's kind of like she's being double rewarded for, you know, that could be like a nice reward system, like in addition to.
Yeah, right. I think I like to highlight that one when I can because it is a really fantastic story. Because she's somebody who'd been using 25 or 30 years, was unemployed, sort of just living life day to day, was somewhat estranged with her daughter and her grandchild. I'm not saying my treatment changed her life, but it seems like something changed about the same time. And things are looking good for her, so that's great. I had another woman who got a job too, hadn't had a job in decades. She got a job at, I think, Walmart and was doing much better. So these are really like, you know, you can pull out single stories when you see a lot of people. That doesn't mean it's all going to work for everybody, but it seems quite promising. And I'm really intrigued to figure out how to do this. So I can have more of these stories. The more people you help, the better.
Great, thank you.
You bet.
I have a question. Do you think there are or know of any like notable differences in patient outcomes in terms of like patients in MAT treatment settings versus other like substance use treatment settings?
You mean like just like groups, or what other treatment settings would you?
So like basically MAT patients versus non-medication assisted treatment patients.
You know, I don't know. I would imagine that there might be some differences. I think the overarching positive thing there is that people are seeking treatment. There's quite a bit of science out there that suggests that people really don't change behavior unless they want to. So somebody who gets over the hump to say, you know what, I need some help. Whatever that help is, they're going to be different than the folks who say, I'm here because I have to be here, right? In fact, the one out of my 9 people who came back from the 4 weeks for the cocaine, the one person who had the least change, or did change, was the one who's like, I don't want to change. I don't care. I'm here for the money, right? So I was able to do something for him. It was reduce his use a little bit, but not a lot. I would imagine there might be some difference. There's some interaction with the pharmacological intervention because it's changing the brain a little bit. I'm not certain that methadone and buprenorphine have similar additive effects with our TMS because they're different pharmacologically. I would be willing to bet that one would pair better with TMS than the other, but I don't know which. But I doubt that they're identical. And with that logic, I doubt that somebody who's on no pharmacological intervention would have the same effect. Now, people who are not on MAT, maybe they have the best effect because they're not already on MAT, and then it scales down from there. Maybe it's the other way. I don't know. These are interesting empirical questions that we need to try to figure out.
Thank you.
I had a question, which is just basically, when can you come to New London to help our clients at our clinic?
I mean, look at my schedule. Like I tell Dr. Podolski, in 2 weeks, my schedule is quite open. So we could schedule anything in 2 weeks. Anything works. I'm happy, Heidi. I could come recruit anytime. Talk to Kim. We're really nice people.
Okay, great. I will.
Anything else? Otherwise, Dr. Steele, thank you for that presentation. It was really wonderful. And it's good to know that, people are doing cutting edge research, with our patient population. I think there's going to be, a lot of good things to come. So thank you for everything that you do. And thank you for sharing what you're doing with our team.
Yeah, it was
So-
my pleasure.